Meet Gertrude B. Elion, known as the architect of cures. At a time when women faced significant challenges in science, Elion transformed medicine through rational drug design. Unlike the traditional trial-and-error approach, she intentionally engineered molecules for specific purposes. By creating purine analogues, she effectively disrupted cellular pathways, targeting DNA synthesis and inhibiting enzymes involved in it, thereby restoring health. This systematic approach led to groundbreaking discoveries, including 6-mercaptopurine and acyclovir, which fundamentally changed the way we treat leukemia, viral infections, and organ rejection. Her legacy is marked by molecular precision. She was a visionary scientist who developed life-saving medicines from the ground up, forever altering the course of modern medicine and saving millions of lives worldwide. Behind every significant scientific breakthrough is a powerful, human catalyst. Gertrude B. Elion's journey into medicine was not inherited, it was shaped by profound personal loss, which solidified her lifelong mission to alleviate human suffering through the power of chemistry. At just 15 years old in 1933, she witnessed her beloved grandfather die from stomach cancer, leading her to make a resolute vow: "I decided that nobody should suffer that much." Tragedy struck again in 1941 when her fiancé, Leonard Cantor, succumbed to bacterial endocarditis, a disease that penicillin could have easily cured if it had been widely available at that time. These heartbreaking experiences transformed her grief into an unwavering purpose to save lives. Despite graduating summa cum laude from Hunter College at just 19, Gertrude Elion faced significant challenges due to a biased academic and professional landscape. She received 15 consecutive rejections for graduate financial aid, with some stating that she would be a distracting influence in all-male laboratories. Undeterred, Gertrude adapted her strategy. She enrolled in secretarial school and took unpaid lab work just to gain experience. She later accepted menial quality control positions, testing pickle acidity, mayonnaise color, and suture strength. Yet, her relentless drive never wavered. By working as a daytime receptionist and substitute teacher, she financed her own education, conducting research at night to earn her master's degree from NYU in 1941. Her determination proved that no closed door could keep her out. In 1944, a wartime labor shortage provided the opportunity that Elion had been waiting for. Dr. George Hitchings hired her at the Burroughs Wellcome Company for $50 a week. Importantly, Hitchings treated her as an equal, allowing her role to rapidly evolve from assistant to full scientific collaborator. This forged a legendary partnership that would change medicine forever. During this time, Elion made a significant decision. While pursuing her PhD at night at Brooklyn Polytechnic, the dean insisted that she quit her job to study full-time. Choosing to remain in the laboratory rather than pursue the degree, she walked away from the doctorate. Although she never earned a PhD, her hands-on work in the lab went on to revolutionize global medicine. Elion and Hitchings fundamentally transformed the field of pharmacology, leading to a significant paradigm shift. Previously, medicine relied on empirical screening, a trial-and-error approach that involved testing thousands of random compounds in hopes of finding effective treatments, often resulting in unpredictable toxicity. They introduced rational drug design. Instead of relying on guesswork, they examined the specific biochemical differences between healthy human cells and pathogens or cancer cells. By identifying particular vulnerabilities, they were able to engineer targeted synthetic molecules, such as purine analogues, that blocked pathogen reproduction without damaging healthy tissue. This method, based on logical principles and detailed blueprints, brought unmatched precision to medicine, replacing chance with purposeful, targeted cures. To combat disease, Elion concentrated on nucleic acids, the building blocks of DNA. She created artificial compounds that closely resembled natural purines, such as adenine, tricking rapidly dividing cancer cells and bacteria into absorbing them. Her team affectionately dubbed this approach the rubber donut mechanism. Just as a rubber donut looks like food but is entirely inedible, this synthetic antimetabolite mimics a legitimate cellular building block used by healthy enzymes. However, when the metabolic enzyme interacts with it, the counterfeit compound disrupts the molecular process. The pathogen attempts to replicate its genetic material, but with its enzymes effectively sabotaged, DNA replication halts, preventing dangerous cell division in its tracks. In 1950, childhood acute lymphoblastic leukemia was almost always a death sentence, with a near 100% mortality rate that took young lives within months. Then came Elion's groundbreaking discovery. By modifying a purine molecule, specifically, by replacing a single oxygen atom with a sulfur atom, she synthesized 6-mercaptopurine, 6-MP. This synthetic compound effectively blocked DNA replication in malignant white blood cells. Later, when clinical researchers combined 6-MP with other maintenance therapies, the survival rate for childhood leukemia soared to 80%. This remarkable achievement in rational design transformed a once fatal diagnosis into a manageable condition and established a cornerstone of leukemia treatment that continues to save countless children today. A single, brilliant discovery can spark a profound cascade of innovation. When Elion analyzed how 6-mercaptopurine, 6-MP, interacted with the body, her keen observations led to a multitude of unexpected medical breakthroughs. First, by noting that 6-MP suppressed antibody production, she engineered a slow-release prodrug called azathioprine. This breakthrough effectively prevented immune system rejection, making the world's first successful human organ transplants possible. Second, recognizing that 6-MP degraded too quickly due to the enzyme xanthine oxidase, she designed a targeted enzyme inhibitor named allopurinol. By blocking the body's overproduction of uric acid, allopurinol became the definitive, lasting treatment for gout. Through rational design, a single molecular blueprint continues to branch out, reshaping modern medicine and alleviating various forms of human suffering. For decades, scientific consensus held that defeating viruses was impossible. Because viruses hijack and reside inside our host cells, conventional wisdom suggested that any drug potent enough to kill a virus would inevitably harm the human body. In 1977, Elion challenged this belief by synthesizing acyclovir. Her brilliance lay in developing a drug that uses an elegant cellular tripwire. When acyclovir is introduced to a healthy human cell, it remains completely inert, harmless, and non-toxic. However, once it enters a herpes-infected cell, it encounters a viral kinase enzyme, an enzyme that is produced exclusively by the virus. This viral enzyme activates the drug, triggering a rapid response that destroys the virus from within, while leaving healthy tissue entirely unharmed. Behold the full majesty of Elion's work, a remarkable array of medical discoveries, each one a targeted approach to alleviate human suffering. By identifying specific biochemical targets, she developed compounds that effectively addressed a breathtaking range of global illnesses. Her synthesized drugs tackled leukemia by targeting malignant leukocytes, combated malaria by inhibiting plasmodium parasites, and fought meningitis and sepsis by disrupting bacterial DNA synthesis. She prevented organ transplant rejection by managing immune cell proliferation, eliminated gout through xanthine oxidase inhibition, and controlled herpes and shingles by neutralizing viral DNA polymerase. Beyond these key achievements, her foundational antiviral methods directly paved the way for her lab colleagues to develop AZT, the world's first approved treatment for HIV/AIDS. In October 1988, Gertrude Elion received the ultimate recognition by sharing the Nobel Prize in Physiology or Medicine with George Hitchings and Sir James Black. Her achievement was historically significant, challenging long-standing academic beliefs. Importantly, she was awarded the prize not just for a single drug but for developing entirely new and rational principles of drug development that transformed medical science. In doing so, she became only the fifth woman in history to win a Nobel Prize in medicine. Most notably, she achieved this highest honor in science without ever earning a formal PhD, demonstrating that a steadfast commitment to research and a passionate dedication to alleviating human suffering can redefine the boundaries of what is possible. Although Gertrude Elion officially retired from Burroughs Wellcome in 1983, her significant influence extended well beyond the laboratory, solidifying her reputation as a lasting global leader and mentor. Taking on substantial leadership roles, she served as the head of the Department of Experimental Therapy, president of the American Association for Cancer Research, and a trusted advisor to both the World Health Organization, WHO, and the National Cancer Institute. However, perhaps her most enduring legacy is her commitment to the future. As a research professor at Duke University, she passionately mentored the next generation of medical and graduate students, co-authoring over 25 papers with them and vigorously advocating for female scientists to ensure that no more opportunities were closed off to women in the field. What truly motivates a medical pioneer? For Gertrude Elion, it was not fame or awards, but an audacious and unstoppable mindset. Embracing the fierce battle cry, "Damn the torpedoes!" Full speed ahead, she confronted every career barrier with unwavering determination. Elion encapsulated her lifelong philosophy by stating, "What we were aiming at was getting people well, and the satisfaction of that is much greater than any prize you can get." This profound empathy was embodied in The Patient File, a cherished personal collection of letters from grateful patients, kidney transplant recipients, and parents of children with leukemia whose lives her drugs had saved. For Elion, this folder containing restored lives was her most treasured possession. True brilliance transcends the laboratory, leaving a lasting impact across generations and shaping the future of human health. Gertrude B. Elion's remarkable journey culminated in significant institutional recognition, including her historic induction as the first woman into the National Inventors Hall of Fame, the receipt of the National Medal of Science, and the award of 20 honorary doctorates. Elion's enduring legacy continues through the AACR Gertrude B. Elion Cancer Research Award, which actively funds innovative, fast-paced research by junior faculty. A notable example is Dr. Peter M. K. Westcott's groundbreaking investigation into the microscopic progression of colorectal cancer, funded by the award. Elion's scientific influence remains a powerful force, ensuring that her relentless pursuit of a cure continues to inspire tomorrow's medical pioneers. Every monumental journey deserves a definitive summary. Gertrude B. Elion's historic legacy can be understood through three profound pillars: her motivation, her paradigm shift, and her ultimate recognition. Driven by personal tragedies, including the painful loss of her grandfather and fiancé, she transformed her grief into a relentless mission to cure disease. Alongside George Hitchings, she engineered a paradigm shift in pharmacology by replacing blind trial-and-error methods with rational drug design, enabling scientists to systematically target biochemical vulnerabilities. This revolutionized medicine and led to her ultimate recognition, the Nobel Prize in Medicine in 1988. She was honored for developing new, life-saving principles rather than just a single drug, achieving one of the highest accolades in science without holding a formal PhD, and forever altering global healthcare.